Hi everyone, this is the second of two blood lectures, and in this lecture we are going to be talking about blood grouping and what it is. So blood grouping was first discovered by Karl Landsteiner, who won a Nobel Prize in medicine for his discovery. Now human beings can have either one, two, or no antigens on their red blood cell. What do I mean by antigen? So antigens are basically glycoprotein molecules. So they are a combination of a sugar and a protein. And they are present on the surface of the plasma membrane of your red blood cells. So it is possible for a person to have only antigen type A, only antigen type B. It is possible to have both antigens A and B. It is also possible to have no antigens on the surface of your red blood cells at all. So a person with antigen A, which is represented here as the circles, their immune system will recognize antigen type A as their own. So anything else other than type A is seen as foreign and therefore attacked by your body. So in a person with antigen A, if you transfuse blood from a person with antigen B, the body does not recognize antigen type B. In this case, the person that is receiving that transfused blood, so a person with blood type A, is going to produce antibodies against antigen type B. Because for this person, A is their own, B is foreign. So their body is going to attack B. So they're going to produce these antibodies, which are proteins in your plasma that attack and try to neutralize threats and foreign substances in your bloodstream. So a person with antigen A is going to produce antibodies against B. So they're going to have anti-B antibodies. Similarly, a person with antigen type B only, their body is going to recognize antigen type B as their own. Type A is foreign to this person. So when they're getting a blood transfusion, if they receive blood from a person with antigen A, their body is going to consider that antigen to be foreign and is going to attack that antigen. So B is their own, A is not. So person B's antibodies are going to attack antigen A. When someone has both types of antigens on the surface of the plasma membrane of their red blood cells, the body recognizes both antigens A and B. So neither one is really foreign for this person's blood, which is why their blood plasma has neither anti-A nor anti-B antibodies. So blood type AB is considered to be a universal recipient. They can get blood from blood type A, and the body will recognize it as their own because they already have blood or antigen A. They can get blood type B, also accept it as their own because they do have B on their red blood cells as well. So that is the reason why people with blood type AB are considered to be universal recipients. Now the last scenario is where a person does not have either of those two antigens on their red blood cells. So this person's body is not going to recognize either type A nor type B. So getting a transfusion from either type A or type B is going to result in their body rejecting the blood cells because they have foreign antigens. So this person's plasma is going to have antibodies towards both type A and B. So they're going to have anti-A and anti-B antibodies, which is why this person is called type O, and type O is considered to be a universal donor because they have no antibodies in their plasma. So because there's no antigens present on these red blood cells, there's nothing that a recipient's blood will attack because there's no antigens present there to begin with. When a recipient receives blood from the wrong donor, there can be a reaction that can cause your red blood cells to clump together. That reaction is referred to as agglutination. In this scenario, the donor has blood type A, as evidenced by these round antigens on their red blood cell. This person's plasma is going to have anti-B antibodies. Now, if this is transfused into the recipient here that also has a type A antigen on their red blood cells, this person also has anti-B antibodies. There are no antigen Bs for these antibodies to attack. So in that case, because this blood donor is also type A and blood recipient is also type A, there is no agglutination reaction. In the next scenario, the donor's blood type is type A, which is these circles on the surface of your red blood cells. The recipient has blood type B, meaning they have antibodies that are going to attack antigen A. So the recipient's blood has antigen A, or sorry, if the donor's blood has antigen A and the recipient has an antibody that attacks antigen A, the blood cells are going to clump and agglutinate. So in an agglutination reaction, this anti-A antibody in the recipient's body is going to attack these type A antigens, and it is going to form a rigid crosslink, which forms little sort of like clot-looking structures, which is bad news because if red blood cells get clotted in your bloodstream, then you can have hypoxia and not enough gas exchange. The next differentiating factor in blood groups is the presence of antigen D, which is called the Rh factor. So when people have antigen D on their red blood cells, which is a similar glycoprotein, they have an Rh positive blood group. Lack of antigen A is Rh negative blood group. So a person can have A positive or A negative blood, or B positive and B negative, same for AB and O blood groups. Now, having Rh positive blood, so having the D antigen present on the surface of your red blood cells is most common. Rh negative is not as common as Rh positive. So if we combine or compound both the presence of antigens A and B and the D antigen, blood group O negative becomes the rarest type. And blood group O negative can only receive blood transfusions from a person that has O negative. However, blood group O can donate blood to just about anybody else. Now, Rh factor is clinically relevant because it can cause a reaction when an Rh negative mother is carrying an Rh positive fetus. So I'm going to explain on the next slide why this is relevant. So here's what happens. So in the first pregnancy, mom is Rh negative, so she does not have antigen D. The fetus is Rh positive, meaning the fetus does have antigen D. So here is the fetal blood, here is the maternal blood. What's going to happen is the Rh positive blood cells here are going to travel and end up in maternal bloodstream. So in the first pregnancy, she develops anti-Rh antibodies because that is the first time that she does get exposed to blood that is Rh positive. So first pregnancy, she's carrying an Rh positive fetus. She herself is Rh negative. So red blood cells from the fetus migrate and travel into maternal bloodstream. And because the maternal bloodstream sees antigen D as foreign, her blood is going to synthesize antibodies against antigen D, or anti-RH as they're calling it here. So she's going to become sensitized to the Rh antigen, and she's going to produce Rh antibodies. In the subsequent pregnancy, the fetus is again Rh positive, the mom is still Rh negative. Now, because the mom already had exposure to antigen D and develops antibodies towards it, maternal blood going through the fetus can cause the antibodies against antigen D, or Rh antigen, to travel to fetal blood and agglutinate fetal red blood cells. And that disease is called hemolytic disease of the newborn. The RhoGAM shot is something that is given to mothers who are Rh negative and have the possibility of carrying an Rh positive fetus. And it's a shot that is given during pregnancy that goes and neutralizes all of the anti-Rh antibodies in the maternal bloodstream. Performing a diagnostic blood test to test blood grouping is actually the opposite of what you would do during a transfusion. So here, you want to induce an agglutination reaction to see what the person's blood group is. So let's call these 3 drops drop 1, drop 2, and drop 3. So testing for blood groups involves taking a finger prick, and 3 drops of the person's blood are placed on this slide. In drop 1, you're adding anti-A antibodies. Let's go over what's happening here. So because there's anti-A, that tells you the no reaction happening over here tells you that there is no agglutination here. So there is no antigen A here for this to react with, right? Which is why you don't have an agglutination or clumping reaction. So no clumping here tells you this is not this person's blood type. Remember, you are trying to initiate an agglutination reaction here. Now, this is an anti-B antibody solution. Here you clearly see clumping or agglutination happening. Now, this agglutination would only happen if the person had antigen B. So antibody B would not react with antigen A, right? It would only react with antigen B, which is why this person's blood type is first B. Now this is antigen B or D, or your Rh factor. The fact that it has agglutinated here tells you that this person's blood type is B positive. So this tells you that this person can receive blood group B positive or O positive as blood transfusions. So I'm going to end here, and that is the end of your blood chapter, and we will discuss some of this and possibly even perform the blood testing in lab.